a really fun day ahead. Hopefully today might be a little lighter in the sense that we're not going to dive really deeply into some new technologies or models, or you're not probably going to see a lot of we're not going to go through any new narratives or download any software. But what we're going to do is, I would say the way I'm kind of viewing this is today is our aspirational day. So today is the day we think about all the things that we've learned over the course of the week. We put them in the context of some science problems that are going to be presented by our presenters today. And then we start to think and dream about what it is we might do with all of the things we've been talking about. And realizing again that the things you've heard about this week are not things that are well developed and finely tuned. They're things that are really on the cutting edge, the bleeding edge of new developments and new science. So it's really great that this community can come together and learn about these things and then dream together about how to improve them and use them to improve all of our science collectively. So we're going to have 4 presentations today by experts in a variety of different application areas. We're going to start with Christof in marine and lacustrine systems. Marco's going to talk about plant soil microbe. Xingyan in watershed systems, and Pamela in wetland systems. And another exciting thing we have that I'm really looking forward to and encourage you to join is the 10:15 session. We'll have a number of our selected students that have been working in the hands-on sessions throughout the week give us some flash talks on their own research and how they envision using some of these tools in their research. And then we'll have a little bit of presentations at 11:30, again, a summary of some resources and opportunities from the Department of Energy's facilities and other resources. So again, just an opportunity for you to learn how to engage further in the future and apply some of the things we've been learning. So I'm going to stop talking and turn it over to Christof. Oh, before I do that, I just want to say, hang in there. Somebody posted this, I think it was Andrea, but if you're feeling that way, hang in there, because I think that we're going to quite soon see some fruit of all of our labors. So Christof, I'm going to stop sharing and ask Christof Meile from the University of Georgia to take over.
Right. Can you confirm that you see my screen?
Yes, I see your screen in presentation mode.
All right. So it's nice start to my presentation with the dog. I hope I'm alleviating some of the pain. It has been a great week. I've actually learned quite a bit, and I'm happy to share some thoughts about modeling of reactive transport linked to microbial dynamics in marine systems. And what I'm going to do today is give a brief start off with recapping some of the things that we have seen earlier in this week, just to highlight a few of the key features about reactive transport models and how they have allowed us to get to a point where we are today and move into the future. I'm going to then switch and present a few applications from my own group looking at methane oxidation in marine sediments under anaerobic conditions. I then will present 2 papers I have nothing to do with, but I wanted to show some work that has been done looking at omics informed processes in modeling water column processes, and then end with some closing remarks. And just to kind of set the scene, I have here a slide that shows some of these features in marine system. This is kind of looking at carbon cycling in marine system, where we have productivity in the photic zone and the surface ocean that is fueling the processes that happen at depth. We have the export of particulate organic matter to greater depth through the biological pump, for example. And this organic matter is then being acted upon or broken down through a bunch of microorganisms, including heterotrophic bacteria, for example. There's the viral shunt that essentially brings back some of that material back into the DUM pool, and so on. So as you can see here, this is a rather complex system that ultimately is also embedded into larger scale food webs. And only a small portion of what is being produced in the surface ocean makes it down to the bottom of the seafloor. This is environment I tend to be working on. And a couple of key questions here that one might be interested in are listed here on the left, related to carbon dynamics, nutrient dynamics, or ultimately the removal of organic matter and burial of reduced carbon over a long time scale in marine sediments. I want to start off with this picture here that looks at the reactive transport model. This is work by Yifeng Wang, who was a postdoc at that time with my PhD advisor, Philippe van Capellen. And this is essentially the figure that Tim already used previously. And it illustrates what reactive transport models are good at, right? So they essentially allow you to kind of deconvolute different processes, how they affect the elemental cyclings. Here, the figure highlights the fate of iron, at least what we thought that was the case back in the mid-90s. So we have the deposition of iron-3 that gets reduced through organic matter mineralization, but also interaction with sulfide, and then reoxidized. And some of it is ultimately being transported back and potentially leaves the sediment again as benthic fluxes. And so we can describe these type of things using reactive transport models as it's depicted down here. And the interesting part really is the reactions. I think, at least to me, the most interesting part is the reactions and the processes that transform different chemicals into each other. So the question is, okay, this was in the mid-90s, where a couple of these efforts has been promoted. There's work from Colin Soutard, there's Bernie Boudreau's work, aside from what I have just shown you. And what are the new things? What are we better at now than we used to be at there? And one of those aspects, I think, is that we have made some progress in the integration of data. And so we have results from models that we can compare with measurements. We quantify the misfit into some objective function, and we can essentially systematically adapt many parameters that typically are involved in this type of modelings to improve how the model results corresponds or matches the measurements. Now, because many of these modeling efforts are relatively rich in parameters, this is a rather important process. And so we have this parameterization that essentially connects the chemistry that we observe with B representing the microbes that essentially drive most of these elemental cycles that we see. So we have tools to deal with that. Now, during this week, we have heard quite a bit about cell models. These are some representations thereof. There's very little I can add to what Hyung Sang already discussed earlier in this week. And these cell models represented here, for example, in the form of FBAs, essentially aim at or provide us some information about cellular activity or growth efficiencies by being informed by omics data. And here on the right, you see a representation of some work that a former grad student of mine have been doing, looking at cell models using a more of a kinetic approach, looking at the kinetics of central metabolism here of Geobacter in particular. The second way where we have really made progress is kind of what was highlighted in the talk by Owen Brody and colleagues also earlier in the week, where we take information about what we know about microbes and relate that to parameters that are of interest, for example, growth efficiencies in our reactive transport modeling effort. The two figures here, this was what he referred to as this codon using bias that Owen discussed earlier. And then I thought it was really nice in terms of what the summer school is, because as soon as he said, I saw on the Discord pop up a few more relationships that people put up with papers that essentially aim at the same thing, taking omics data and relating it to properties that are central to reactive transport models. So the second thing that I think we have made progress on is illustrated here is that because we have better computers, we are actually able to resolve a couple of things that we couldn't do in the early days. And one topic that is of particular interest to mine is shown up here. So this is a picture from a paper by Eric Christensen and colleagues. And what it shows, this is in a sediment here, so a couple of centimeters, and you see this U-shaped structure, which is the remnant of, this is a burrow of an organism. And you see, hopefully, that, you know, the coloration here between the sediment and surrounding the burrow is distinct, which is indicative of distinct redox conditions associated with these burrow structures. This is also reflected in some work that Niels Folkemon has been leading, and he has used planar optodes. Here I'm showing you a result from one that measures oxygen, and this is again, we are looking at a cross section in sediment. You can do that by essentially using ant farms, so thin aquaria. You put one of these planar optodes on the side and you take pictures, and these pictures really reflect the variation in the concentration field of oxygen. So here we have an anoxic sediment. Down here we have the oxygenated overlying water, strong transition because it's a carbon-rich environment, but then you have this feature, and this feature is associated with a bivalve living down here and moving water around that leads to the intrusion of oxygen into an anoxic sediment. Now, this is here a spatial feature, but this is also something that is highly dynamic. These animals pump and move water around. So on this axis, you see time. This is over an hour. This is a cross section here through this shown here, B, and you see that there's oxygen present. Oxygen has been used up, present, and being anoxic again in between. And the white line here is a measure of pressure and an indicator of the pumping activities of these organisms. Now, and we are now able, or have been for a couple of years now, able to relate these observations to modeling efforts. And I think the beauty here that I wanted to highlight is that we have a limited number of chemicals that we can measure like that. So for example, oxygen, but you know, we can build on that, and using a reactive transport model, we can essentially expand on that and make predictions that are informed by actually some data up here, make these predictions for other chemicals that might be of interest, for example, related here to the nitrogen cycle. The third thing that I think we have made quite some progress on is making these codes more user-friendly. And I know that may seem like a stretch to some of the people who are not doing this for a living, but I also want to stress, I mean, you saw that during this week, I mean, the ability to run really complex simulations very, very quickly, I mean, is to me quite staggering. And this has been highlighted by a number of talks that have been given earlier this week here in the context with Floatrans, some of the web interfaces, or the sandbox that is part of that. I myself have been involved in some other efforts where that essentially couple the symbolic expressions that you introduce into Maple. So you can basically write down reactions more or less as what you do on paper, and you can integrate that in an automated fashion and create corresponding Jacobian matrices and so on that are directly used to then do simulations in some other complex reactive transport models. And I think this is important because it really allows us to be linking our work more closely to some of the stuff that also you have seen earlier. So this is some graphs out of the Kelly Wrighton's lab, and as you have seen using DRAM, we can take information from sequencing efforts and essentially then link it back to and infer functionality in the environment. And so here we can adapt our reaction networks and account for here the absence of say disammonium nitrate reduction to ammonium. Okay, so this is in a nutshell kind of a recap of some of the work that I think is important that facilitates this microbial reactive transport models. I want to switch gear now and talk a little bit about work that is my lab was involved in. And as I said, I want to talk about anaerobic methane oxidation in marine sediments. And just to give a little bit of context here, now we are looking at the concentration profiles of sulfate, methane as a function of depth in a marine sediment core. You see the depletion of sulfate here, and methane essentially is only present below the zone where sulfate has been depleted. When you do rate measurements using sulfate that is labeled with 35 sulfur, you can essentially measure that these regions, transition regions, are places where we have high rates of sulfate reduction. So the geochemists have told us for literally decades that this is essentially what is happening. And about 20 years ago, then the microbiologists here, a paper by Antti Boetius and I think Kai-Uwe Hinrichs was involved in that, looked at what they found there. They observed these aggregates consisting of archaea and bacteria, and they inferred that these aggregates were involved in this process of anaerobic methane oxidation. We have looked at that from a modeling perspective, and we looked at it in a couple of different scales. It is kind of highlighted up here. We have done some work really at the large scale, asking, looking at in a more classic reactive transport modeling sense, what are the dynamics and the environmental conditions that you would observe here in a seep site. And so this is work by Julian Rose. And what he did is essentially looked at quantified some flow in seep environments and both seep and non-seep environments in the Gulf of Mexico using radium as tracers, and then coupled it with reactive transport models that accounted, for example, for methane oxidation, and then the production of this leads to production of alkalinity. This alkalinity then stimulates the formation of calcium carbonate. This calcium carbonate has an impact on the permeability, and so he looked at how does flow dynamics of fluid flow dynamics, what they are expected to change over time. So this is kind of the environmental setting. We also looked at it at a very, very small scale, okay? So focusing on these aggregates themselves. And this was motivated by some work that I had done with Beth Orcutt when she did her PhD with Mandy Joy. And we did some modeling of this type of microbial aggregates. Now, a few years ago then, we teamed up with Victoria Orphan, and her lab really had done some quite interesting things because they looked at these aggregates at the aggregate scale. They looked at their activity distribution using nanosims, so looking at the nitrogen uptake as within aggregates, and they also looked at their genomes. And one thing that they encountered is that what they found is that these multi-heme cytochromes were rather abundant in some of these organisms involved in this here, highlighting these enemy groups up here. And so what they suggested based on this is that the interactions between the bacteria and the archaea in this microbial consortia is not necessarily linked to the diffusion of dissolved electron donors, but maybe something that is more looks like that, direct interspecies electron transfer. And a grad student of mine, Xiaojia he teamed up with great Chadwick from Victoria Orphan's lab to model this type of systems. And what you see here on the bottom right is kind of the data that we were using. So this is the fish data. You see the archaea and the bacteria in different colors in here. And then this is the N15 measurements that were made in the Orphan lab using nanosims. So we essentially took this type of data, cell densities, arrangements, and environmental conditions, and some of their incubations, combined it with some work that we found in the literature describing extracellular electron transport. We put this into a small-scale reactive transport model accounting for thermodynamics of these reactions as well. And we essentially, our model simulation supported the idea that, that molecular diffusion is not fast enough, and that in order to explain this type of activity patterns is really something we need to have faster transport, and for example, related to this direct interspecies electron transfers. And so this is something that we looked at, and some of George's work shows that quite nicely. Now, so I talked about, work that happened at the really large scale here, kind of looking at the environmental conditions, some work that happens at the aggregate scale. But I want to focus now on something that kind of happens in between. And this is work by Hee-Won Young. He's also here in the summer school. And I'm sure he's going to be happy to talk to you guys all about this in more detail on the Discord server. But what he did is essentially he established a pore scale model that is kind of depicted here on the left-hand side. It's the lattice-Boltzmann modeling approach. And with that, you can essentially simulate, solve Navier-Stokes equation, so simulate the flow here in pore channels. And we coupled that. The next step is to solve an advection diffusion reaction equation to simulate the dynamics of chemicals and biomass. And we use this for a couple of applications highlighted down here. And I want to highlight just the first one here, which we use this type of models of the pore scale to address the question of how much does it matter if you're not resolving the pore scale. And so he did simulations under a different range of flow and reaction conditions, looking at reactions where a substrate is being used up by microorganisms that are distributed somewhere, at discrete locations within these modeling domains. And so explicitly doing this type of simulations, he then found that was able to quantify the how bad are we doing if you're not resolving the pore scale for these particular types of reactions. And so here is kind of an outcome of these type of simulations. The details are in a recent paper that he published. What you see here on the different axes, this is the Dam-Kohler number, which shows the how important is reaction relative to advection component. And on this is a Peckley number, which gives you a measure of how important is advection relative to diffusion. The vertical axis is a measure of the misfit. Okay, this is what we refer to as R1. This is the naive estimate of reaction rates if you only look at bulk properties, and we compare this to the actual volume average reaction rates if you're resolving these pore scale simulations. And you see that depending on the flow and reaction conditions, you get a variation of about up to a factor of 4 or something like that for this type of reactions that we were studying here. We also found that the actual distribution of the microbes is maybe a little bit of a lesser important, at least under the conditions that we studied. We also looked at a few other things, looked at communication between microorganisms in the context of quorum sensing, and then feedback of biofilm growth on the transport properties in the porous media. I'm not going to talk about these things. I'm sure Hee-Won is happy to chat with you if you're interested in that. But I do want to go back to this last part here, is connecting it back up to this metabolism, because this was one of the key motivation for developing this type of models. And so in the project that supports this type of work, this is supported by DOE Genomic Sciences, led by Victoria Orphan. Chris Henry is part of that project as well. And you heard him talk earlier about some of these things that they're working on. So they are looking at to build these flux balance models for some of these archaea and the sulfur reducers that we are really interested in the context of AOM, right? So we have a connection with that. And after hearing his songs talk about yesterday, I think we will certainly go and look at how we can speed up some of these simulations. So again, I think this is a really nice part about the summer school. I mean, you guys get exposed to some cutting edge work that I think has really great potential as you move forward as well. Okay, so in a nutshell, some of this work that I have shown you here, I want to show that we use reactive transform more in a classic sense to provide some context in space and time. We can use it to look at interactions here, the work that we did with the aggregates. And I think what is really important here, and particularly in the context of the summer school, is that we can use these models kind of to connect scales and link up microbial metabolism, particularly to the post scale and then ultimately to larger scale, which was shown in Hivon Young's work. I want to switch gears here and talk a little bit about work of other people. So I hope I don't butcher that, but I think it's important because it kind of gives you a little bit of a broader perspective. And there is actually, after I put this together, I found this paper by Hellweger, really nice summary that essentially boils this down. So if you're interested in this, go maybe start here. That gives you a good overview, and then before you dig into some of the individual papers that Ferdi Hellweger is discussing. And I'm going to highlight two aspects here. One is looking at nitrogen and sulfur cycling in oxygen minimum zones. And the other one looks at connecting ocean biogeochemistry with some trait-based modeling. And just to set the scene here, so the first example, we're looking at the oxygen minimum zone. So these are environment that you find, for example, in the Arabian Sea over here, in the eastern tropical Pacific, both north and the south here. And these are environments where the oxygen in the water column basically goes down to essentially zero. And it's quite exciting in a sense because, as you know, when you run out of oxygen, the biogeochemistry, gets, how should I say, interesting or more interesting even. And particularly when you look at some of these processes when it comes to nitrogen cycle, denitrification, NMOX, discriminatory nitrate reduction to ammonium and so on, but also linked to the sulfur cycle shown here. Okay, so what Dan Reed did together with Christof, Julie Huber, and Greg Dick is they essentially studied one of these environments. They looked at the water column profiles in the Arabian Seas, used data from a paper by Pitcher and colleagues from the NEOS that they collected on a cruise a little bit earlier. And the way they went about looking at this system is as follows. So they built a reactive transport model that involved the organic matter mineralization linked with oxygen, nitrate, nitrite, sulfate, and so on, okay? So in addition to that, then they accounted for aerobic oxidation processes involving some of these reduced species, sulfate and ammonium in nitrification and so on. And then finally, they also considered NMOX and sulfide oxidation with nitrite. And so you can do this, you can essentially put this into a reactive transport model where you solve, the chemical dynamics due to the transport processes and then these reactions that I've shown here. The transport in this case was basically parameterized based on water column stability measurements, so relating to the observations in salinity and temperature profile. Now, nothing new here, I would say, but what they did then is they essentially introduced marker genes for these different processes that are listed in here. And they essentially looked at the temporal evolution of these marker genes as well, subject to transport, reactions, and then also, basically a decay or a death term that removes these genes again. And then they simulated this reaction rate as a function of, growth parameters, uptake parameters, processes, depend on the availability of substrate. the machinery present to process these substrates, some inhibitors, and then the energetics. I'm simplifying this slightly, but that's kind of the gist of what they did. And the good news is, I mean, they were able to reproduce the main features in the concentration profiles here. This is profiles now not in the sediment, but in the water column. So we have about 2,000 meters water depth. We have oxygen. The dots or the markers here, these are the measurements, and the lines are their concentration, simulated concentration profiles. And so the main features, something that they represent, were able to reproduce. But also, I think more importantly, is that they were able, their model kind of mimic the distribution of these marker genes, as it is highlighted here for ammonium oxidation here, or Animox features here as well. And then they looked also and kind of related the production or the dynamics in these genes, and they basically related that to the messenger RNA as well. Maybe there's another way is they essentially, you know, the model led to results that was consistent with the understanding of what's happening in the Arabian Sea, so denitrification is exceeding Animox as a process to produce N2. Now, so you could say, well, you know, you've probably seen better fits, and what's the big deal? And I think what was really nice about that work is that it's kind of illustrated in here. Now, it was a setting where there was no sulfide was detected in the system. And you can essentially have a couple of ways how you get to that. First of all, you may not have production of sulfide, so no sulfate reduction taking place in the system, or you may have a system where you have sulfate reduction taking place, but then the sulfide that has been produced is being reoxidized quickly. And using their model, what they were showing is that when you look at these two scenarios, for many of the chemical profiles, they look relatively similar. There's some difference here in the nitride profile, where one may argue that maybe this is a distinguishing feature between these two scenarios. But what is really striking is that the model produces a very, very different distribution of genes, okay? And so using these genes basically as a biomarker of indicator of processes. And that is essentially what the people have seen experimentally. This is a paper by Don Canfield and colleagues, where they looked at this very question at the different site. This is an oxygen minimum zone here of Chile. This is the concentration profiles that you see here for, here oxygen in blue, in the top 200 meters of the water column. So you're running out of oxygen around 60 meters. This is a place where then they find high sulfide oxidation rates. And they also saw evidence of significant abundance of genes that are involved in active sulfur cycling. Okay, so first application here. The second one that I wanted to briefly talk about is how you can use omics or connecting it to looking at phytoplankton community distribution. And this is work that is, the groundwork has been laid by Mick Follows and Stephanie Dukiewicz, and they essentially establish a setting. And again, you have seen similar picture like that, I think, in Owen's talk earlier in the week, where we looked at the information about this based on the genetics and the physiology of organisms. You combine them with the environment, you have basically a competition between different organisms that have different traits that leads to emergence of an ecosystem structure and function. And this was quite successful. They actually were able to kind of reproduce some of the key features of the biogeography, community structure that was arising, and they were essentially selecting for organisms that are, I would say, similar to some of the important organisms that we see in nature as well. Now, Victoria Coles and colleagues, including some from my own department up here, built on this work, and they essentially looked at a situation across the Atlantic. They used a physical oceanography model using HICOM to simulate the transport of and the flow of water. They combined that with a model that accounted for about 70 microorganisms that differed in size. There was a random selection of what these traits were being. There's size-dependent substrate affinity and so on. But then these organisms could do a bunch of different things. So they had about 20 different biochemical functions that could be selected for, that included, can they use light? Are these nitrifiers? Can they move around? And are they nitrogen fixers? And so on, okay? And they essentially simulated these organisms with having these different traits here. And these traits were linked to, again, to some what may call marker genes. And they looked at what was emerging in terms of community structure and in terms of the chemical signatures, as well as the gene distribution and the transcription levels that were associated with these simulations. So again, using a natural selection approach, if you're not successful, if you're one of these 70 organisms and you're not successful, you got replaced by another random seeds and so on. So the system is basically allowed to evolve. And here is the key result, I think, from their feature was that they did a bunch of different models on, and the emergent metabolic function of the ocean biogeochemistry was relatively stable. And there's kind of, what this shows here is kind of the model domain that is shown up here. This is the distribution of genes in the models in the background, and the circles are the observational data. Comparing, this is a measure of the transcripts here, and again comparing the simulation results with the data here in circles. Okay, and so they found a reasonable correspondence between the model and the measurements. They found that the similar metabolic functions would be expressed in different model runs, and again, these are randomly seeded, so important finding. But what they also found is that the emergent microbial community actually was quite different. And I think in a nutshell, what that means, it's really that we had an emergence of different microbial communities, but they're kind of doing the same thing. And from a modeling perspective, that's actually quite helpful because it really allows us to focus on some of the functions. And the first part here, I want to stress again that I think here the key take-home message is omics that sees things that are not apparent in the chemistry, and we can use this in models. Now, there's not much here in terms of the metabolome, so I wanted to briefly switch gears with regard to that. So we have heard quite a bit about that in terms of how we can link organic matter, knowledge about organic matter, to its reactivity, and that goes back to some earlier work, shown in here. I don't have time to go into this, but there's other paper here by Shannon Banner that kind of illustrates really that the molecular properties are, as the title says, a primary control on the microbial utilization of dissolved organic matter in the ocean. And you have seen this graph before quite a few times, the work by Doug LaRoe and Philippe van Capellen, that relates the characteristics of the organic matter here, nominal oxidation state, to the energetics. Okay, and so there's a great potential, and you saw that before by bring this all together, looking at the lambda theory or looking at this joint microbial DOM characterization studies that are ongoing, and AMSL is doing a great job in that area. I do want to discuss a little bit more about that in terms of the environmental controls, because I think this is important. There's a couple of factors in the environment shown up here. Is temperature. This is work by Nat Wesson and Mandy Joy, and it really shows the temperature can change the relative importance of different pathways. Now, I do think that while this is a complex system, this is sediment slurries, sediment from coast of Georgia, but these are still complex systems, right? But I think the tools that we discussed today are probably helpful in helping us understand some of these dynamics. Things get a little bit more, I think, more complex even is when you start to look at, the interactions of minerals with the organic matter. This is some old work, by Kyle, Meyer, our Ransom, and so on. And there's a lot going on in this area, also in the terrestrial environment. But I want to focus here on this last part a little bit. And what this shows is that This is work by Eric Christensen again that showed that the organic matter pool in sediments that were either defaunated or had Arenicula marina, one of these worms that I mentioned earlier, can be hugely different, right? And so I think I'm mentioning this is because we can have these additional controls that are not microbial, that both shape the dynamics in the systems, as well as can also shape the microbial community. This is work by Longhui Deng that showed essentially, looking at a bunch of different sites here between Sweden and Denmark, essentially showed that the microbial community, both archaea and bacteria, are quite distinct in the non-bioturbated setting from the bioturbated setting. So the elephant or the hippo in the room here is that we can have important top-down control as well on the processes that we are studying, and we have been very much focused, and for good reasons, on the microbial aspect as well. Now, this is important for a number of reasons, and I think I'm basically kind of running out of time, if I'm not mistaken. But I did want to stress that there are other ways to kind of look at these process, not just as a bottom-down approach, but also looking at in the context of overall systems approach. And this is some work by Joe Valino. I think I'm going to basically skip through this, but I'm happy to talk about that. And the key here is that we may need to, when we have really complex systems, we may need to think about ways that allows us to capture some of this complexity, not just by adding in more and more and more microbes, and because we also need to account for their interactions and so on. And I think that's something that Pamela Weisenhorn probably going to talk about more about in this afternoon. But we need to take into consideration the need for also look at some other organizational principle in the context that may be arising in the context when we have food webs as a whole, not just microbial systems. Okay.
Christof, it's 9:12 a.m.
Okay. So again, these are approaches that can also be linked to omics data. But so I do want to basically end with this and leave a moment or time for discussion, is that kind of what we saw this week is that we can have these emerging properties that result from the micro scale, and I think it's really important to also constraints look at the macro scale. But I think what is really promising these days is the combination of different data types, both on classic concentration distribution, but then connecting with different omics times, an understanding of the organic matter, and so on. I think this is, to me, is really, really exciting. It's a huge challenge, but exciting. I wanted to bring home really the usefulness of models to make some of these connections across these scales that you're seeing in here. And I think I do want to stress again, the challenges that you're facing to really account for some of the complexity and the interactions between microbes themselves, between not even talked about viruses at all, but also in terms of relationship to upper trophic levels and so on. Okay, and with this, I just want to make sure that I acknowledge the people again that are involved or have been involved in some of this work. Jörn Rose, I showed some work from him, seed modeling. AOM work that was done in my group by Xiao Xiaohe, and then Yang's work is partnership. The data comes from Victoria Orphan's lab here. In particular, I had involved Sean McLean, who is now in Tokyo, and Chris Campus. And then the work on the bioturbation is the stuff that I have been doing together with Niels Wolkenborg and Ian Dreyer. And then the microbial connections here with Mark Lever's group at ETH. And here Longhui Deng was the lead author on that study. And I think that's all I have for today. I appreciate and I'm here to answer some questions if there's time.